Okay, alright, so we're just going to record this first part just so we can record the video for biology. Let me double check, see if we have to go over anything else. No, we will be covering a quiz later. If we have time after the biology lab, we'll do a fun Jeopardy game quiz that will be on chapters 1 through 5 for our lab for quiz coming up. And if we do that, we'll film it. For now, we're just going to watch the video, then we'll stop filming, we'll move over to the microscope and actually start filming the experiment. So here we go.
Although other non-plant cells can also have vacuoles, plant cells have these characteristically gigantic vacuoles that are largely a storage place for water. While animal cells are around 70% water, plant cells can be 90% full of water, and they need a place to put it. However, in addition to water, a vacuole can also contain food and other nutrients, as well as waste products. The central vacuole can also contain digestive enzymes like those in animal cell lysosomes. A central vacuole can also help maintain a neutral pH in the cell by pumping hydrogen atoms, or protons, from the cytoplasm into the vacuole. What happens if you pump a lot of protons into the vacuole? This makes this plant cell component acidic, which is also similar to a lysosome. So how does the cell wall in the central vacuole relate to those plants you forgot to water? Remember that cells transport water across their cell membranes through this process of osmosis. When you forget to water the plant cells are then in a hypertonic environment, or an environment with less water around them and more solutes. This causes water to leave the plant cells. The consequence? A wilting plant. Notice, however, that even in a hypertonic environment, the cell wall helps maintain the shape of the plant cell. As soon as you started watering the plant regularly, the plants were again in an isotonic or equal water environment. As a result, the cell's central vacuole takes on more water, and the cell can fill the cell wall's shape. Now, what if you poured on a little too much water to try to make up for lost time? You may have seen a plant wilt when it isn't watered enough, but have you ever seen a plant explode with water? I doubt it, and that's because in a hypotonic environment, with more water outside the cell, the central vacuole will swell with water, putting pressure on the cell walls. However, those cell walls will keep the cell rigid and strong. It'll prevent the cell from bursting. This pressure that is put on the cell walls is known as Turgor pressure. In fact, most healthy plants are in a constant state of Turgor pressure, and this is what gives plants their rigid shape.
So,
in summary,
is it bad or good to overwater a plant?
I think there gets to a point where it's not, and I'm not going to rely, I'm not sure of the scientific reason for why. I think you can't overwater a plant, but yes, the plant does have certain protections that if you overwater a little bit, it's going to be fine, generally speaking. I think it does depend on the plant, depends on the cell structure.
We've learned more about two important plant cell components today. The first is the cell wall, which is a protective layer surrounding the cell on the outside of the plasma membrane. The cell wall...
I'm going to pause it real quick. This is one of the things we're going to be looking for in our lab today, because when we cut up the onion, we look at it, we're going to look for all these ridge lines and see if we can see where the cell walls are. So we should be able to catch it.
It's composed of cellulose, or a polysaccharide sugar that provides strength to the cell wall.
Okay, if you want to, you can copy that down. So that's one term you need to know, cellulose. So essentially it's the sweet substance that provides strength to the cell wall. And this will be the only video we have to watch for biology, so yay. We'll have to be working with our hands after this. I also brought onions that I grew from my very own Publix.
My mom grew some from her own garden. Just real quick, everyone does have their lab manuals ready, right?
way yet?
No, I had it myself.
Oh.
A second structure important to all plant cells is the central vacuole, which is a large storage compartment in plant cells.
That is also very important. You don't have to write these down, but I just recommend these. These are all terms you're going to want to know for reasons. I'm not going to say why. So the central vacuole is a large storage compartment in plant cells. We're going to do a quick PowerPoint after this video. I'm not going to spend too much time on it. But just essentially just so you can kind of see what you're looking at when you're looking at cells, you'll be able to see all the individual pieces. So a central vacuole should not be confused with a vacuum, which is also a large storage compartment, depending on what size you buy.
Who has a vacuum that's a large storage compartment?
I think some big vacuums out there. Scientific discussion.
Both the cell wall and the central vacuole help plant cells regulate water movement, or osmosis, to keep plant cells strong enough to withstand a few days without water.
Okay, yeah, that was it. Okay, so short thing, and we're going to move over to the biolab real quick. We're going to present this. Okay, so some of the things you're going to be looking for in terms of our onion is to see if you can spot the nucleus. It's going to look like these little divot-shaped things. The cell wall, which is going to look like these veiny membranes running throughout. The cell membrane, which is actually going to be the the layers in between these lines, and then the vacuoles. Now the vacuoles are a bit hard to spot. There's these pockets essentially. So we're going to move over and we'll see a little bit more of an illustration of what this will look like. Okay. So in this regard, we're going to learn how to prepare and do a stain slide. This is going to be a little different from what we did last time. I think we're going to try it once without the stain and once with, but we're going to do a wet slide first, look under the onion just like what we did with the hair, and then we're going to try to put one blue drip of, I can't remember what it's called, but essentially blue coloring, and we're going to be able to see like all the little layers and stuff.
I did that at home a few days ago.
Good. You might be able to help me out then, because we'll see.
I did a purple onion wet melt slide for my siblings, and I looked like leather.
Nice. Well, that actually is good. You've got more experience than I do on that. Alrighty. So, the main things you're going to be looking for, some of these we will not be looking for at all. We just won't even be able to see them. But one of the main things we're going to be trying to identify is the cell wall, which is pretty easy. It's not hard to spot. And we're going to try to identify chloroplasts as best we can. We may not be able to see them in these, but we will hopefully be able to see them. So that's one thing you want to know is what chloroplast is, and we can look that up later. Essentially, chloroplasts are little tiny nutrient-based, well, I don't know if it's nutrient-based, what you call it, but a little, what's the word I'm looking for? Chloroplasts are like little chloroplasts containing chlorophyll where photosynthesis takes place. Now, can anyone tell me what photosynthesis is? Yes.
Changing? Like,
kind of.
Okay.
You are right, because it is synthesis, it's the process of change, but what is happening? What is the plant getting, first of all? Like, what is it trying to get?
Food.
Nutrients and energy, right? And it gets that from the sun, from light. Yes, photosynthesis. Alrighty. Okay, and we're also going to be looking for the large central vacuole, which again is kind of the big sac that's like in the middle of the cell. We're going to be looking for that. And I think that's about it as far as this goes, yeah. So, but this is just for the benefit of the remote students that this is what we're going to be looking for as far as that goes. Alrighty. With that being said, we will stop recording. You can just push the button, the red button there.